but I actually don't mind if I don't know. I don't mind trying to know. Guys, can we not do
I think the first part today is getting through and after that, you get a score. When is that? Yeah.
Then you meet where?
First thing I'm going to recap today is what we covered last week, from primary to tertiary structure. Okay, so for those that felt like you weren't able to draw the link between all of that, we're going to do a recap. After that, we're going to go to the next level of structure. Yes, there's one more. Oh, everyone done secondary math? Recap. So I'm going to recap primary, secondary, tertiary structure. Then I'm going to go to the fourth level. And then finally we're going to complete CA1.5C, which contains denaturation. Okay, that's questions D and E. Let's see, is it okay?
Yeah.
Alright. Started already?
Yeah.
Alright. This is for practice. Okay, so today we're going to do a quick recap on the 3 levels of structure that you can observe in proteins. Let's begin with the first level of structure. That is the primary structure. How would you define a primary structure? You can discuss with your table mate. How would you define a primary structure? Okay. May I hear from Mani? How will you define a primary structure? Different types and arrangement. Okay. Different types. Okay. Rather arrangement, more of like a sequence of the amino acids. And perhaps you want to include a number as well. Right? If we change these 3 factors, actually the number of primary structures we can create is infinite. Yeah? Okay, so when we refer to primary structure, we're referring to the number, type, and sequence of amino acids in the polypeptide. I've had students ask me, why would you consider number, sequence, and type as a structure? Right? Often when we think of structure, we think of something very physical. We can see a difference in shape. Right? When we look at the secondary structures, we can see that there's either helical, alpha helix, or beta feeder sheath. Right? Why would we consider this a structure? Well, you can think of it as a structure from a certain point of view. Imagine I have different amino acids joined together. Okay, let's just work with this first versus this. In a sense, you could say it's a structure because at the end of the day, the entire polypeptide, the shape is also different. Right? If you look at it in its entirety, the shape, right? If I were to just do it this way, I'm going to erase away the amino acids in a while. You'll find that actually the shape of the entire polypeptide, when you look at the primary structure, is different. I don't think I can erase properly, but you get it up. Okay, so in a sense, there is a difference in structure. Okay, if I just look at this polypeptide below and the polypeptide above, okay, the structure is quite different. Of course, right now I'm just using shape to represent. What would make a shape different would be the R groups, right? Every R group has a different structure, different shape. Putting them in a certain sequence will result in it's like a different skyline of a different city, right? Every primary structure is like a different skyline of towers. Okay, so that's the primary structure. May I ask, what's holding the primary structure together? What bonds are holding the primary structure together? Okay, it's a polypeptide, therefore held together by peptide bonds. So peptide bonds hold the amino acids together. At every level, let's talk about what kind of interaction sequence. Yeah? Let's go to the secondary structure. How will you define a secondary structure? Okay, I heard something from the back, Shanice and Keller.
Alpha helix and beta sheets.
Okay, there are two kinds of secondary structures. If you want to really describe what a secondary structure is, I would say that they are, okay, in a sense, they are localized folds and they have patterns, and the two kinds of patterns, the alpha helix or the beta sheets. They are localized folds, and as Yuan was mentioning, we see a pattern. There are two kinds of patterns. One of the patterns as mentioned by Shanice is alpha helix. The second kind of pattern we see is beta pleated sheets. And we say that they are localized folds because you don't find like alpha helix throughout most of the time. You find them, at localized regions along the polypeptide. I cannot remember if we did this.
Yes,
We did. Okay, therefore, my next question is, what's keeping the alpha helix from just entangling into a primary structure again? Hydrogen bonds, okay? So, hydrogen bonds. But how will you describe where the hydrogen bonds are? I could say it is between, right? The hydrogen bonds are between the CO group and the NH group of peptide bonds. Okay? More specifically, the hydrogen bond will be between the oxygen and the hydrogen. Yeah? Again, why? Goes back to our first lesson. Okay, the idea that they are polar. Last but not least is the tertiary structure. Okay, I'm going to take a pause right now for you to take down any notes you want to take down. But if you are done, could you discuss with your friend how you describe a tertiary structure? What are some key things? You see this protein, it has a tertiary structure. How will you describe a tertiary structure? Okay, number one, you see that it's highly folded. Anything else? Okay, it has a 3D, you could say globular shape. Okay, I would say globular shape is not a definite shape per se. It just means it looks like a lump. But a bit weird to say that tertiary structure means you see a lump. Okay, so it has a globular shape. What's holding the entire polypeptide, you know, folded, highly folded and staying in this 3D globular shape. Okay, there are interactions and bonds involved. Okay, 4 you can mention. One is ionic bonds. Okay, hydrogen bonds.
Hydrophobic interaction.
Thank you. Sorry, you say what?
Hydrophobic interaction.
Okay, hydrophobic interactions. Last but not least, yeah, the disulfide linkage, which is a form of covalent bond.
What does it mean when you say a form of covalent bond? Like, is it a covalent bond? Or
Oh, sorry. It is a covalent bond. Sorry. Yeah. But it's just like how a peptide bond is a covalent bond. Yeah. So I'm just saying that if someone were to say that one of the kinds of bonds you can use to hold a polypeptide together is covalent bonds, they are specifically referring to a disulfide linkage.
how does it happen? Like, is that condensation reaction or?
From my research online, it's not exactly a condensation reaction that brings the two SH groups together. It is some sort of a oxidation reaction that I think Mr. Low will teach. It has to do with removal of electrons and hydrogen ions in order to achieve that. If you want to go one step further, the ionic bonds occur between charged R groups. Okay, not RGPS, charged R groups. Okay, hydrogen bonds occur between polar R groups, yeah? Hydrophobic interactions between non-polar R groups. Disulfide linkage between, specifically, the R groups of cysteine, which looks like a SH, right? Their R group contains a SH. Okay, this should be very, very specific. Yes.
And for the hydrogen bond, you say it's between the O of the CO group and the H of the NH group is is it of different amino acids or in the same amino acid?
Oh, okay. It will make sense if it's a peptide bond that is further away. Yeah. So for example, we look at this one over here. In order for this helix to hold its helix form, right? You will expect that the hydrogen bond is between this oxygen and NH group that is further away.
So not in the same amino acid.
Yeah, it doesn't quite help with anything if, let's say we have a CO and then NH right beside, right? This is a peptide bond, right? Bond together also like not quite meaningful, yeah? So this hydrogen bond needs to occur across peptide bonds that are quite far away from each other. Okay, in order to hold this spring structure or the beta Peters. I see?
When you get up to 16 together, right, is that mass to form disulfide bond? Can just stay at several people?
Oh, is it a mass? Good question. I would say in biology and in chemistry, usually things it's not a mass, it's usually if the conditions are right. So I would assume that in the case of this particular reaction, I think for it to happen well, it may require certain, what we call pH, certain acidity. Often oxidation or reduction reactions require a certain kind of acidity or maybe some temperature for it to happen. So I won't say it's a mass. But in the context of, let's say, a question, and you see two cysteine groups nearby, you could assume that it is a possibility to form.
So is it possible for like the bonding to be between amino acids?
Sorry, how do you
like in the amino acids, like the O bond to H right below it, instead of the other one.
So your question is, how is it possible this
Is it possible
Oh, is it possible that an amino acid bonds
to hydrogen? I mean like hydrogen bonding between, within amino acid.
So are you saying like, what do you mean within an amino
Like
acid?
just now Shanice asked.
Like that
Like this, is it?
Yeah.
Is that
Oh,
possible?
okay. Oh, is this possible? No, it is possible. But what I'm saying, it doesn't contribute to the shape.
So when we are asked to identify hydrogen bonds, we don't draw that.
I would say drawing this doesn't answer the question. Yeah.
So there's no point in drawing that. They are looking for the
Yeah,
hydrogen
okay.
bonds between
Yeah,
different
okay.
amino acids.
Yeah, because if you have a helix, it will make sense that the hydrogen bonds need to be across, or rather a distance away, to hold this string structure together. If I have the hydrogen bond just beside, let's say a O over here and an H over here, just hydrogen bond like that, it's not contributing to the shape. Okay? But does this happen? Of course, I cannot say no. Yeah. Yes?
I think also to ask whether the hydrogen bonds, identify the hydrogen bonds that doesn't affect the shape.
Oh wow! I mean, I don't know. But anyway, I would like to say that when I share with you ways in which we could assess, it's not I'm not trying to be exam-centric per se. Yeah, I just want to show you the possibilities so that you can open your eyes a little bit more. But we try not to be too focused on what can and cannot come out, because then it will limit me. Right? If I say this cannot come out, then I cannot be creative anymore. Okay? So keep your mind open. Now a simple activity. Could you draw a protein that only has primary and tertiary structure? No secondary structure. Okay? Protein number 1. Okay, protein number 1. I'd like you to draw a protein, right, using just lines. Okay, you can draw squiggly lines with primary and tertiary structure only. And then draw a second protein with primary, secondary, and tertiary structure.
What does it mean when you write like 1D, like the little
Oh, yeah, it's just my short form writing primary and tertiary, secondary. It's easy to write the whole thing up. Okay? Often I like to do this just so that we can see if we understand the different levels. Yeah. Oh, what would it look like, right, for protein to have primary and secondary only, no tertiary? Try. Yeah, I showed before last week, is it? Yes.
For the joint, what do you mean? Like, we need to
Are you asking if we ask you to draw anything more? Depends on whether the structure is alpha helix or beta. Okay, and for anyway, this is for beta, that is for alpha helix. That is for beta helix, like one string go down, then go up, then go down, go up. Alpha helix is this one. So the context of the question. localized means that the fold is not very extensive. It's found within a fixed region. So if we look at this structure in particular, this fold is one case is only found here. Yeah. Let's say no. Yeah, it's possible. So like what you've done now, this is localized. It is local in this region. Okay, ready? What would a structure that only has a primary and tertiary structure look like? The protein. So for example, this. Imagine this squiggly line represents the polypeptide. Okay? So primary and tertiary structure, yeah? Tertiary structure because it's highly folded, globular shape. What if we have secondary structure now? Okay, level 2. I would include some secondary structures inside. Okay, let's include an alpha helix here. Let's include perhaps a beta theta sheet over here. Let's include maybe another alpha helix over here. Now I have a protein with primary, secondary, and tertiary structure. Okay? What about a protein with just a primary and secondary structure? What would that look like? Straight. Keller, can you draw? Yes, you can. Okay, so I shall invite you up. Yes, Keller can draw. But Keller, may I see what you are saying? If you demonstrate what you're saying. Hey, you still have a skateboard. Quick, Keller. You want to skip up to the front? Keller, I need to help you. Okay, Keller, help me do protein with just a primary and secondary structure. Draw where? Okay, draw here. Okay. Go. Go. Thank you. Okay. Primary, and we see two forms of secondary structures, but we don't see a tertiary structure. It's not highly folded. Yeah? Okay. Now, let's try real examples. Let's search for real examples. What protein should we play with today? Let's go with. Have you done keratin before? I felt like we did last week. Yes.
So you know the hydrogen bond he was doing. Must he label the positive and partially positive, partially negative charge?
I mean, look at the context of the question. Yeah. I will always do my best to label everything so that we always go back to our roots of why we do things. Yeah. Maybe in year 4 I'll stop labeling. So and then I will assume you know why there's a bond. Okay, so let's look at yes. Oh, this is a C, yeah? Yeah. Yeah, you're right. O and O cannot, O and O cannot. Okay, proteins. Trypsin. Ready? Let's see how many levels of structure has. Trypsin, protein, structure. Trypsin. Trypsin is a protein that breaks down proteins? Yeah. Oh yeah. Wow. It's like an inception thing. Okay, a protein that breaks down protein. Let's look at this. How many levels of structure does this protein have? 3. All 3. Okay? How many types of secondary? 2. Okay? This is the skill to pick up. Let's try another protein structure. Okay, I remember on the what makes COVID-19 as a virus so potent is because of the proteins present on its membrane surface. It can cling onto our respiratory system very well. So let's look at that protein. COVID-19 spike protein. Wow, cell. Okay. Okay, let's look at this here. Wow. What level of structures do we see here? All 3 can? Yeah? Okay, but at this point I'd like to bring up the 4th level of structure. Notice that this spike protein consists of actually 3 polypeptides interacting with each other. If you ever see a protein that is color coded, for example, here we see green, pink, and blue, it means to say that there are 3 different tertiary structures or 3 different proteins side by side interacting with each other. You may be wondering, how are they interacting with each other? If I just put 3 proteins side by side, won't it just fall apart? It's interacting using these same 4 bonds. Yeah? Okay, so I give you an example. For example, let's say I have protein number 1, and then I have protein number 2. The amino acids at the edges can interact with the amino acids at the edge of the protein also, of protein number 2, yeah? Via these same 4 types of bonds. That's what's holding more than one protein together. So, what is a measure of a quaternary structure? If you observe more than one protein interacting with each other. And you can tell that by the many colors here. I searched this last week, but now let's look at it with different eyes. Okay, now that we know that it goes up to quaternary structure, we can analyze each protein a bit better. For those that are done, I'm going to press enter. Let's look at hemoglobin as a protein. What level of structure does it have? Oh. Okay. Sorry, so you know what is the tension you're feeling here?
No, is it like the red one? Is the red one on the left connected to the red one on the right, or are they two?
Oh, okay. Okay. Lilian is asking, since you only see 2 colors, does it mean this red one over here is a continuous structure, and the blue one is a continuous structure behind? Okay. The reason why it's colored the same is because this protein over here is exactly the same as this one. In reality, there are actually 2 of the same things interacting with 2 of the same thing. Okay? Hemoglobin as a protein actually consists of 4 separate proteins stuck together. But there are only 2 types of proteins. Okay? So sometimes they may show you a protein that looks like this. 4 different proteins. Sometimes they may just show 2 colors because 2 of each of the proteins are actually the same, exactly the same. Yes?
Then in that diagram, how will we know whether it's like connected or not?
Okay, so it is not whether it is connected or not, it's whether you can tell that there is more than one protein present. Okay? Yes?
What if the 2 polypeptides are identical, so it's the same color?
So for example, let's say this protein is made out of, this protein has a quaternary structure, made out of 2 proteins, but each of the units are exactly the same, right?
Then what if the color is the same?
You're right. usually in such an instance, I would say, I think we will still represent it with two different colors, just so that you can tell.
But he will say that it's identical.
We may say that it's made up of exactly the same primary sequence. Okay, but you know what's the issue? We're poor, so we cannot bring things in color. So you will see things in black and white. So what does this mean for you? Okay, so there are two ways about it. Usually we will shade each protein a different shade of gray. Yeah, so you can still tell. Okay? Or sometimes we will deliberately make it very clear that there are two separate proteins and then we'll put it very close to each other. So you can tell that they are not one continuous shape.
Will they touch? Or is it just
Actually they should touch as they interact with these bonds. Yes.
I thought it's quaternary structure is more than one polypeptide, right?
Yes.
Then you wrote more than one protein.
More than one, okay, because the definition of a protein is a folded polypeptide.
Yeah.
Yeah. So actually I could say more than one polypeptide, more than one protein is okay.
So a quaternary structure may consist of more than one protein. Of course, there's many.
So is it a protein?
Okay. Okay. So it's okay to say protein or more than one polypeptide, both are fine. Because sometimes you will see people describe each one of these as a protein subunit. Okay?
So each one of those colors, is it a polypeptide or is it protein? Or is it, can it be counted as both?
I would say that you could use both. Which will be more comfortable if you see polypeptide?
Yeah.
Yeah. Because perhaps to you right now, when we say protein, we're looking at just one whole, right? I think that's fair enough. You could use the word polypeptide, maybe we are more comfortable
So
with
then
that.
we describe the entire structure as one protein or do we?
So you can describe the entire structure as one protein made up of 4 separate polypeptides. Or sometimes people will say this is one protein made up of 4 protein subunits. Yes?
Is it quaternary or quaternary?
Oh, okay.
Quaternary.
You say again? Is it?
Is it quaternary or quaternary? Because you wrote it differently in that one.
Quarter. Oh,
Quaternary.
quarter. I think I'm missing a few alphabets, is it? Quarter. Okay, quaternary. Thank you. They are spelled wrongly also. Quaternary, okay. Yeah, quaternary. Usually if you see a marker that doesn't, I tell my class people. Yeah, sorry? Yeah. Okay. The bonds that hold 2 separate protein subunits or 2 separate polypeptides together are the same bonds that will hold a tertiary structure together. Okay, let's pause a while. I'll just take as much questions as possible. Yes. Oh, what are these green things? What are they? If I'm not wrong, it is a pigment found in hemoglobin. Yeah, it is the pigment. It looks like the pigment. The thing that actually will hold on to oxygen. It's a pigment or protein. May or may not be. Some pigments may just be chemicals.
What is a pigment?
What is a pigment? Pigment is something that can give color. Like chlorophyll is also a pigment.
So there's pigment in this protein, right?
Okay. Can the word polypeptide be interchangeable with protein? I would say, if I were to read online, sometimes people use it interchangeably. If you really want to go to the nitty-gritty of it, a polypeptide doesn't imply folding at all. Okay? In a sense, yeah, if I were to say this is a polypeptide, I'm essentially saying this is a string of amino acids. But usually when people say the word protein, it implies that it could have several levels of folding. So in other words, a protein is a folded polypeptide. To say that a protein is not a polypeptide also a bit weird, because a protein is a polypeptide, it's just a folded one. Yes?
They're not held together by peptide bonds. That is that one section is one protein
You mean like this case?
Yeah, like all at like then this one is one.
And then there's another one that's interacting with it.
Yeah, like this the polypeptide is 5 by 5.
Oh, yeah, yes. Okay, a polypeptide will be defined by the peptide bonds that are holding this bunch of amino acids together.
Yeah, so this hydrogen bond don't count.
At least for a single polypeptide. Okay, we'll take a short pause here. Okay, you can consolidate everything first. Then we try one more protein. Anything else that we know of, there are proteins that we want to look at? Have I shown you the red cube protein yet? Yes.
But if you have two of the same protein, right, what do you differentiate with tertiary or primary?
If I put two of this exactly the same protein side by side,
Yeah.
how do you tell whether it's interacting with each other or not?
How do you tell if it's tertiary or quaternary?
Okay, how do you tell if they are just simply two proteins with tertiary structures, or it is a quaternary structure that consists of two proteins interacting with each other? Is it? Is a question? I would say to a quaternary structure, the two protein subunits or the two polypeptides will be very close to each other.
If you just have a lot of the same protein, is it a lot of tertiary structures or a lot of, or just one entire quaternary structure?
Okay, why do you think very fast when you're speaking? You said that if there are a lot of tertiary structures
If
together,
there are a lot of the same protein,
Yeah.
then how do you tell if it's a lot of tertiary structures or just one entire quaternary structure?
usually if there are proteins that are very, very close to each other, it will be quaternary in structure because there are likely bonds holding them close to each other. If not, they can very well just fly away.
So the entire thing will be one quaternary.
Yeah, so there are a lot of proteins that are made out of many, many subunits. Let me show you one. This protein is called Atp Synthase. You ready?
What does it look like?
Okay, look at this protein over here. This is considered one protein. Made up of many, many, many protein subunits, or many, many, many polypeptides. It has a quaternary structure. So when we say more than one, wow, it doesn't stop at like 3 or 4. It can be really, really big. Okay, this is one of the largest proteins you can find. It is more specifically an enzyme also. It is found in our mitochondria. It is what produces the energy, the powerhouse of them. You always call it powerhouse, right, mitochondria. This produces the energy, the mitochondria.
And for example, if you just have two of that thing,
Oh, if I have two of these side by side, would it be considered a quaternary structure?
then it will be one entire thing or would it be two?
Yeah, I don't understand your question. Say again.
Like if you just have, because there's only have one of this, right?
Okay.
Then if you have a
Versus?
lot of them together, then you have a lot of quaternary structures or is it just one entire structure?
Oh, okay, I say again. You hear? Let's say you're asking if I have one more of this, right?
You just have a lot.
You have a lot of them.
Yeah.
Okay, I say again, a quaternary structure, the proteins need to be very close and interacting with each other.
Yeah, then is it multiple or is it one?
Okay, so you're asking this, is this a quaternary structure? Is that your question? Versus, what I'm telling you is, we can only consider something a quaternary structure if they are closely interacting with each other.
How do you know it's closely interacting?
You look at this, they are like stuck to each
Yeah, but
other.
if you have a lot of them together.
Okay, so you are saying, what are you saying actually? Are you saying that if we have like that, is it?
I think a lot means like more than 2.
Then
I
how
got
do you
like
tell
10.
if it's if you have the same one?
Okay, let's use this example. Okay, I draw this structure. That's what's on the board and over here. Okay, are you asking if I have many of this, what structure is it?
Yeah, like you have a lot of them.
Okay, but I'm saying again, you need to see whether they are interacting very closely with each other. Okay, if they are far apart, they are not considered one protein by itself. Yes.
Then would it be possible for like those two that you draw like be one quaternary structure even though it's made out of like 2 of them?
In this case, I don't think so. I don't see these proteins interacting with each other, these large protein structures interacting with each other. They're normally far apart. They're quite far
Is it
apart.
possible to have like 2 quaternary
Yeah. I mean, okay, if I were to cut this into half, right, would you not say that this bottom half is still a quaternary structure because it's made out of many different polypeptides? The top is also a quaternary structure because it's made out of many polypeptides. So as long as there's more than one, it's going to be called a quaternary structure. Don't care how many there are. Yeah? Yes.
So you're saying all those bonds in a tertiary structure also can be found in the quaternary structure between polypeptides?
Yes. Yes. No, there could be ionic bonds holding the two large proteins together also. Yes. Say again. If it's a what? Quaternary structure. I would say this protein has a quaternary structure. So there's no need to say that there are many quaternary structures. As long as there's more than one polypeptide, we see as a whole is a quaternary structure. So okay, look, if I have a protein that's made up of two subunits interacting with another protein made up of three subunits, I won't say that there are two quaternary structures. I would say this thing has a quaternary structure. Yes.
So in theory, if you put like two of these very close together, then they will be considered as one quaternary structure.
You could say that if it really interacts with each other via all these bonds, yes, it becomes one large quaternary structure.
I think asking how do you know the difference between closely interacting and not closely interacting?
Okay, again, it goes back to really they are touching each other. Yeah? They will be touching each other. So, how should I say it? They will be touching each other. No, but because they are still made up of different polypeptides, the polypeptide doesn't connect head to head, amino acid to amino acid. Yeah. A bit baffled. Why? Okay. I've had many wonderful, fruitful conversations with my sim in the past 2 years or so. Okay, ready up? The very last part. Okay, denaturation. Okay, when we talk about denaturation, we know that our proteins are held together, the structure is held together by a variety of different bonds by now. Okay? If you look at question D, the question is asking you, if I were to put something in a very acidic environment. Okay? So look at this question over here. Okay, what if I put it in an environment that has a lot of H+? Okay, a lot of H+ ions. So I'm going to zoom in right now. Imagine now I zoom in to these two amino acids. Okay, here is the carbon, here's the carbon. Here is an R group, here's an R group. Okay, they are interacting with each other. Okay, let's say this R group, both of them, they are charged R groups. So perhaps positive and negatively charged. So this is an ionic bond. What if we submerge this protein in an environment that has a lot of H+ ions? Okay? Yeah, what will happen? One of the things that will happen is that, okay, so imagine this is a very good relationship. Okay, between the plus and the minus. They are attracted to each other. But then suddenly I introduce a lot of like extra H+. It can come in the way of this relationship, yeah? Who will this H+ be attracted to in this relationship? The possible the negative ions. Yeah? So imagine all of these H plus starts to get attracted to the negative ion, the negative charge group over here. Yeah? And what going to happen is that it disrupts the ionic bond. It's like a third party, like just come in, spoil this relationship which was the ionic bond. Ion dipole interaction. What? This is a this is an ion. This is also an ion. Is there an ion-ion interaction? Oh, then it's a new kind of.
But isn't like the same type? Is it when like dipole is something the thing comes in and
Yeah. I mean, I don't know what exactly this kind of interaction is. Is it considered ionic because there are two ions? Yeah. I'm not sure if it's considered ionic. I don't know the terminology of it. But one thing's for sure, the H plus ions will be attracted to the negatively charged R group. It could disrupt the ionic bond. With? Oh, like H plus and this, right? Yeah, okay, I don't know if this is called ionic bond. Perhaps it is. Yeah? Because after it's two ions, by definition that's an ionic bond. Yes?
The ionic bond, right, when like the plus and the minus is like attracted to each other already, wouldn't the entire thing become neutral?
Oh,
So then it wouldn't like attract anymore.
okay, but that's not the nature of ionic bond. Ionic bond, yes, there are two oppositely charged things, right? But overall it's neutral, but those two charges still exist. Okay, overall it's neutral, but those two charges still exist. And therefore it can still very well attract other charges if it's present. Yes?
Does this apply to all the other bonds?
Okay, so then we can explore that. Let's look at a hydrogen bond. A hydrogen bond, technically, the H plus ions could disrupt the hydrogen bonds by being attracted to the partially negatively charged oxygen. It could disrupt hydrogen bonds too. Could it disrupt things that are non-polar? Nothing much to disrupt because H plus ions aren't really attracted to non-polar R groups. What about cysteine? Not really, because if we form a covalent bond, overall it is non-polar as well. It shouldn't be able to attract any H plus ions. So if I were to submerge a protein in an environment with a lot of H plus ions, these two bonds can be disrupted. Yes?
I was asking about what bonds you mean.
Okay. These two bonds will be quite severely affected. Yes?
So is this how like the pepsinogen is activated by HCl to become pepsin?
Or you're asking, is that how, say again, what becomes pepsin? Pepsinogen becomes pepsin because we put it in H plus, is it?
But then there's like all the other enzymes don't work well with acidic.
Right. Okay, so if you notice every time we introduce H plus ions, it can affect two kinds of bonds. Hydrogen or ionic. If I were to remove the bond right here, it would mean that this part can unfold. Right? So it's kind of related to what you're asking. In the presence of different amounts of H plus, how the protein fold will change slightly. So that's how it can be activated from pepsinogen to pepsin. Because now we change the shape a little bit, become activated. For those that are a little bit lost, we are going back to GH2 enzyme. Yes?
Then it's not possible for the introduction of like H plus to break the actual peptide bonds, right?
Right. So you cannot break peptide bonds, that's a covalent bond. Ions will break or interrupt or disrupt bonds that fundamentally are built upon a foundation of opposite charges. Okay? You could, if I can technically extract out all the H plus again. Yeah. So what this means something, technically I can destroy a protein level of folding just by splitting it with a lot of acid. Yeah? Yes?
So the ionic bonds and hydrogen bonds get affected. So does it mean that they will break?
Yes?
So they will break, and so the protein will be less stable.
The protein will, the folds that were held together by these two bonds, right, will start to unfold.
Also unfold, so the shape will change.
Yes, the shape, overall shape should change. Okay, so if I were to introduce a new vocab today, denaturation is the idea that proteins can unfold. When we say a protein denatures, it means that it loses its shape as the protein unfolds. Yes. Okay, so I use the word disrupt because technically, if I have very little H plus, this ionic bond may not be fully broken or disrupted. If I have a lot of H plus, you expect that the interaction between the opposite charges won't be as strong as before. So different amounts of H plus you add can disrupt to different levels. Yeah. So say this say this, what do we call this, a pipe cleaner. If this is a protein that is once globular in shape, if it's held together by a lot of ionic and hydrogen bonds, let's say I submerge it in an environment with a lot of H plus, it will start to unfold because all the ionic or hydrogen bonds get disrupted. Yes? If the outputs are non-polar, then chemistry is not affected. Yeah, so it could be some areas are still folded because maybe this part is interacting via a covalent bond. So the areas that are held together by the ionic hydrogen will be affected. Yes? It really depends on how extreme this is. Yeah? There's one way, one sure way that we can cause this protein with a tertiary structure to unfold completely. And that sure way will be E. Okay, question E. What if we heat things up? If I heat things up, we will gain kinetic energy, vibrate more. Actually, the vibrations can be strong enough to sever not just the ionic hydrogen, hydrophobic interactions, covalent bonds also can break. If the temperature is high enough and the entire polypeptide vibrates to the point, all the outputs start to shear apart. Yes?
Like when you and put quaternary structure into acid, then it can become separate tertiary structure.
Possible. And then again, those tertiary structures could unfold some more and you lose the tertiary structure, left the secondary. To unfold some more and all we're left with is the primary structure. Yes? Yeah. Same, the other way around also. Because you haven't learned acids and bases yet, have you?
You're learning right now.
Oh, you just started. Okay, so Mr. Low, if we talk about bases as OH minus, right? OH minus serves the same effect. If I have too many OH minus, you'll get attracted to this positively charged one. I thought you haven't learned yet, so I only focus on H plus.
So it means that the ionic bonds will also break.
Yeah.
So the same thing happens, but basically it's on the positive ones.
Yeah. Yes. Sorry, Yishin and Hayu. Yes. Oh, okay. Very kind of you. Okay, so now Tang is asking, can the heat break the peptide bond? Meaning individual amino acids start coming off, right? Okay, possible? I wouldn't rule out. If you heat it up to the extent of, it's possible. So it could happen. Okay, yes? Okay, you're going to ask that. So you notice that when you cook an egg, what you start off with is a very translucent thing, right? You know egg white is made of proteins? Yeah? It starts off very translucent, right? That's because the proteins in there are all globular in shape, sliding over each other, and light can still pass through them. But the moment you start to cook an egg, you start to cause all the bonds to break, and it starts to untangle and untangle. And when you have enough untangled proteins, they start to intertwine with each other, such that it cannot move anymore, becomes solid state. Intertwine to the point light cannot pass through, becomes opaque. That's what actually is happening when you cook an egg. You're actually denaturing all the proteins. And this entire mesh will start to get intertwined with each other. Can you uncook and make that all? If you somehow can take out each strand, and then somehow you can get them to refold themselves again into their globular shape, then you could uncook an egg.
How does the invisible part of the egg when you cook it become white color?
Or as I was saying, when you start off with, when the concept of color means it reflects whatever you does not absorb, right? So if it's white, means it does not absorb any light. Am I right? Right, white color. Physics students. So you're asking how it becomes opaque, right? Okay, so imagine lots of small proteins just sliding over each other like liquid state. Light can still pass through the gaps between the proteins that are sliding over each other, not really interacting with each other. But when you start cooking them, they start to untangle, and they start to form a mesh that is just intertwined with each other. Then it becomes solid state now, because now it cannot flow. And the more mesh you have, the more you cook it, less light can pass through. Yes. Sorry, yes. You're talking about the egg yolk, is it now? Oh, okay. Egg yolk membrane. Why does it? Okay, that's because you know what the egg yolk membrane is made up of? It is made up of lipids. Okay? It's not made up of proteins, it's made up of lipids. You know, like a cell membrane. Cell membrane after all is lipids, right? Yeah. So you cannot denature lipids, but you can denature proteins. I think
Cell
we are
membrane
overrun in DM time.
Cell membrane is
Yes.
a lipid.
Yeah, cell membrane is made of phospholipids.
Oh, the one that can
Yeah,
let it
remember?
pass through.
Mm.
So
Okay.
denaturation is how do you even pronounce it?
Like how you just did.
Denaturation.
Yeah.
Okay.
So proteins can denature under extreme acids or bases, or under extreme temperature. These two instances we could denature proteins. Yes.
So like proteins will only like serve their purpose best in neutralized environments and not acidic or basic.
Therefore, yes, proteins have an optimal condition. If their optimal conditions are not met, they suddenly become too acidic or too hot, then it will lose its shape and function.
So
Yes.
each protein's ideal temperature is different. Like ideal acidic
Right. Each protein's ideal temperature actually is different. You know the enzyme in your mouth, the ideal temperature is of your mouth temperature. The enzymes in your stomach made of protein, the ideal temperature is also the stomach temperature. The ideal acidity of amylase is whatever is in your mouth. The ideal acidity for a protein enzyme is actually very acidic in your stomach. So every protein has its own ideal level of acidity or temperature.
Wait, then what other
We're
things?
jumping so many chapters. Yes.
What other things can be
I shall cut
denatured?
Sorry?
What other things can go through the denaturation?
Just proteins.
Oh, okay.
Yeah. If you denature something, what you're left with is a primary structure. No folds anymore. Because the folds were held together by all these bonds which were broken. Yes, I
Is there
see.
any way to break only the covalent bonds?
Yes, say again.
Is there any way to break only the like hydrophobic interactions or covalent bonds?
What an interesting thought. Therefore, I shouldn't use acids, right? Correct?
Like
Okay,
break
so
only the covalent.
I don't know if there's a way to isolate only break the covalent bonds. I would suppose the only way to break it would be to find a chemical that targets only that bond. Yeah. A chemical that only targets that bond. Or maybe an enzyme that targets only that bond. That perhaps is the best way. Okay. Wow.
So if the amino acid R chain is non-polar, then it's not affected
By acids?
by
Yeah.
So it will just remain at the same shape if it's all non-polar.
In theory, yeah.
What if there's some non-polar but some polar?
Then perhaps the level of it being affected won't be that great.
So you're saying that part of it will be opaque, but part of it will also be able to let light pass
you're talking now as a single protein or many proteins?
As a single protein with different like polar and non-polar.
Yeah, actually, as a single protein, light we wouldn't really consider whether light can pass through. Okay. Yes. Got that? Let's go. Let's stop the recording.